good. So we got the tools here. Nice. They specifically examined over 5,000 stone tools from a Chateau-Perronian site called, oh god, all these names, I don't want to say it all. 45,500 years ago, which is about 5,500 or so years before the last Neanderthal presence. Comparing these tools to other sites in the region, they identified that the culture did not derive from older regional Neanderthal technologies, such as the Mousterian, but rather from elsewhere. This suggests that Neanderthals who were making Chateau-Perronian stone tools migrated to the Iberian Peninsula, possibly from France. The Chateau-Perronian culture did not come from Homo sapiens either. Instead, older Neanderthal populations were replaced by the Chateau-Perronian Neanderthals who occupied Iberia until 43,000 years ago, who were then replaced by Homo sapiens. This is an interesting study because these patterns in local Neanderthal cultural shifts provide a more granular view into prehistoric life. I welcome these kinds of publications as they change. Okay. Well, that's interesting. So, you had the native population of Neanderthals there completely replaced by ones from France, and then eventually Neanderthals died off. Yes, the Iberian Peninsula was kind of the last stronghold for the Neanderthals when they were slowly dying out. Alright, let's move on. Okay. After the Neolithic, European population showed an increase in height and intelligence, reduced skin pigmentations, and increased risk of cardiovascular disease due to genetic changes that lowered concentrations of good HDL cholesterol. The changes reflect ongoing evolutionary processes in humans and highlight the impact the Neolithic revolution had on our lifestyle and health. Write researchers from Nijmegen and Hanover in Frontiers in Genetics. Research of these past events offers interesting starting points for today's science and healthcare. Just like plants, animals, and other organisms, humans are dynamic organisms and variable traits. Look at how humans behave and their appearance, and you will see differences in skin color, eating habits, susceptibility to diseases, height, and so on. Such external features are called phenotype. This appearance can be influenced by, for example, genetic factors, social and cultural habits, eating behavior, and environmental factors. Nijmegen scientists in, maybe it's an H, Nijmegen? I don't know. From the Helmholtz Center for Infection Research have investigated whether they could trace the development of some complex human traits of modern humans from, say, 50,000 years BC to the present. These are complex traits such as height and intelligence. In this case, complex means this trait is not determined by one gene, but by dozens or even hundreds of genes. Each of these genes has only a very small effect on such a trait. I'm kind of bored with this. Moving on. New non-destructive DNA method opens opportunities. A new method of obtaining ancient genomic data without damaging source material has been developed by University of Otago, researchers creating new opportunities for museum and archaeological collections worldwide. Ancient DNA doesn't have to be destructive, says lead author Lascaux Scarsbrook. Our new method allows to the reconstruction of genetic wakapapa without, what is that, wakapapa? Without destroying the very bone that's kept its secrets for thousands of years. Wakapapa genealogy, a fundamental principle of Maori culture that is critical in establishing one's identity. Ah, okay, so it's genealogy, but in Maori language. The research published in the International Journal Molecular ecology presents a new method of obtaining genomic data from small vertebrate remains that causes no visible damage to the underlying bone. This will not only facilitate analysis on materials in museum collections that are either too small to be destructively sampled, but also more rare and valuable materials, both culturally and scientifically. Newly sequenced DNA data allow researchers to understand and show how tectonic activity, climate change, and human impact have influenced. Okay, I want to know how they did it. While South Island populations show genetic breaks, geographically, significant loss of genetic diversity in both North and South Island populations is testament to the impact of humans and introduced predators. It's previously thought that you could only distinguish between bones of different gecko species on the basis of size, but amazingly, CT scans and ancient DNA showed that we could, in fact, distinguish between geckos using shape alone. Size got thrown out with the bathwater. It turns out size doesn't matter after all, meaning that we know about New Zealand's geckos at this time of human arrival is now a paleontological clean slate currently. Okay, we first attempted to obtain mitochondrial genomes using a different method, and after months of time in the laboratory, we failed to produce usable data. After going back to the drawing board and making some changes, we achieved our goal, which just goes to show that perseverance, what is it? Long-term conservation of finite specimens is of huge concern to curators globally. So you didn't say. The beef of your freaking article is not about this fucking technique. It's about the history of geckos in New Zealand. That's not what we want to know. Has been developed creating new okay, whatever. Moving on. New study sheds light on early human hair evolution. Hair is an important feature of primate, including human diversity and evolution, serving functions tied to thermoregulation, protection, camouflage, and signaling. However, the evolution of wild primate hair remained relatively understudied until recently. Researchers in the Primate Genomics lab at the George Washington University examined what factors drive hair variation in a wild population of lemurs known as Indridae. Specifically, the researchers aimed to assess the impacts of climate. You can't hear? Can't hear me. Can't hear me. What do you mean you can't hear me? Hello? Can you hear me now? I hope you can hear me now. You should be able to hear me now. Oof, that is really behind. Really behind. Why is the stream so far behind? Well, I guess I'm hoping you can hear me. Oh, God. Okay. Well, let's hope you can hear me. Okay, you can hear me now. Okay, let's continue. Talking about lemurs and their hair. Specifically, the researchers aim to assess the impacts of climate, body size, and color vision on hair evolution. Sifaka lemurs, which are native to Madagascar, have denser hair in dry, open environments. The researchers believe that, like early humans, the lemur's hair helped protect against the strong rays of the sun. Lemurs in colder regions are more likely to have dark hair. This is the first evidence in mammals for a classic pattern in nature called Bogart's rule, which states that dark colors could aid with thermoregulation as they help absorb heat from the sun's rays. Red hair in lemurs is associated with enhanced color vision. According to the researchers, populations that can see a larger range of colors are more likely to have patches of red hair. If only that was true with us. I have supervision. I'd like it. Okay. Multiple evolutionary pressures may act on one trait, and the strength of their influence may vary between species. Human hair evolution remains a mystery largely because hair does not fossilize. Lead author on the paper and postdoctoral scholar at the University of San Diego, California, the lemurs we studied exhibit an upright posture like humans and live in a variety of ecosystems like early humans, so our results provide a unique window into human hair evolution. Brenda Bradley, an associate professor of anthropology who directs GW's Primate Genomics lab and is a co-author on the study, explained our understanding of hair evolution and diversity in other primates helps us fill in the gaps of our own human evolutionary story. Most people are intrigued by the diversity of hair on their own bodies and the varieties of hair types among people around the world. Bradley said understanding our hair patterns in non-human primates such as these lemurs may provide a comparative context for understanding how variations arose in human hair. Yeah, diets. Talking about the farming stuff. Yeah, it changed us a lot. I don't know what's up. My stream, because I can see it here, is really far behind what I'm doing. I don't know why. Don't know how to fix that. I'm sure it'll be okay. Alright, moving on. That explains our lower back pain. Anthropologists turn to Neanderthals for answers. Examining the spines of Neanderthals, an extinct human relative, may explain back-related ailments experienced in humans today. A team of anthropologists have concluded a new comparative study. The analysis centers on the spine's curvature, which is caused in part by a wedging or angling of vertebrae, and the intervertebral discs, the softer material between the vertebrae. Neanderthals are not distinct from modern humans in lumbar wedging and therefore likely possessed curved lower backs like we do, explains Scott Williams, an associate professor in New York University's Department of Anthropology and one of the authors of this paper. However, over time, specifically after the onset of industrialization in the late 19th century, we see increased wedging in the lower backbones of today's humans, a change that may relate to higher instances of back pain and other afflictions in post-industrial societies. A good part of this perspective derives from the wedging of Neanderthals' lumbar or lower vertebrae. Their spines in this region curve less than those of modern humans studied in the US or Europe. However, much of this view was based on an analysis of modern humans beginning in the late 19th century, well after the onset of industrialization, which significantly altered our daily lives. Furniture, for instance, became more widely available and desk jobs were prevalent, both of which encouraged sitting and with it changes in posture. These changes were coupled with reduction in high-activity occupations such as agriculture. In addition, specific afflictions became associated with working conditions that elicit poor posture. Past research has shown that higher rates of lower back pain are associated with urban areas and especially in enclosed workshop settings where employees maintain tedious and painful work postures, such as constantly sitting on stools in a forward-leaning position. In other words, by examining spines from humans who lived in the post-industrial era, past researchers may have mistakenly concluded that spine formation is due to evolutionary development rather than changed living and working conditions. Okay, well, like I said, bones are much more malleable than we think. Overall, they found that spines in post-industrial people showed more lumbar wedging than did those in pre-industrial people. Moreover, Neanderthal spines were significantly different from those in post-industrial people, but not from pre-industrial people. Notably, the scientists found no difference linked to geography within samples from the same era. Pre-industrial versus post-industrial lifestyle is an important factor, explains Williams. who acknowledges that because lower back curvature is made up of soft tissue, the intervertebral discs, not just bones, it cannot be ascertained that Neanderthal lumbar lordosis differed from modern humans. Lordosis is that curve that you see in the bottom of the spine. So there's actually a couple different curves going on. Right over here for you. Let's see. Make sure it's actually being seen. Oh, there. Not a very. It's a little low. Just gonna get a better view of Gregor here. There we go. So you have Gregor, my little helper. The lumbar lordosis is right here. So right here you have your lumbar. You only have about 5 of them. And then you have your thoracic and cervical. And you have this massive curve here. And they're saying that Neanderthals and ancient humans had pretty much the same curve. It's when we started actually sitting around and working more desks and chairs, and maybe even not that, but just having our modern-ish amenities post-industrial revolution caused this to increase, so we're having even more so of this curve, and it causes a lot of back pain. It's the curve of the spine having this kind of S shape going on is what makes us be able to walk upright. It's one of the things. There's multiple things that had to change, including the pelvis becoming much shorter and wider, angling of the femurs as well. But yeah, that's what they're talking about. And if you look, so Gregor here has some like rubber discs placed in, and they are quite thick. You need a lot of cushion going on there to be able to get that spring when you walk and everything, so that everything isn't too jostled around and broken and things like that. And they get much smaller, thinner. And when you have in like remains and such, the stuff like arthritis, you can find that in the lumbar bones really well compared to other vertebrae. Okay. Continuing on. So that's interesting, and it just goes along with the farming and the height is that our modern lives, and by modern I mean since like farming is so new, we really don't know everything it's done to us yet. Having such a complete overhaul of your life, your daily activities, it's going to have a drastic effect on everything having to do with your body. And we don't know what all that farming has done. We don't know everything that modern jobs have done, and that since the Industrial Revolution, we sit around on computers a lot more. I mean, we didn't have computers in the early post-industrial times, but we are going to see some drastic changes to us, and it's not an evolutionary change, because I'm sure also if they wanted to take this data and compare it to any hunter-gatherers out there, this would probably show a difference as well, and it's due to our daily lives. Our bones are much more malleable than people think. I can look at some bones and tell you quite a bit about an individual and their daily lives. You can also tell perhaps how muscular the individual was. I had one ilium, the blade of the pelvis here, right here, And all of this is where the glutes attach. And you can actually see where the attachments are. So you have this ridge here, that's a really good attachment. This ridge is another one, and this one. I had one individual whose ridges were so well defined and prominent and sharp almost. They had to have had some really good muscle attachment there. Buns of steel. Anyways, yeah, your bones are living just like the rest of you. They are harder to change, but our daily lives, we don't know what this is doing to our health and long-term health. It'll be interesting. Maybe we'll all end up looking like the, the Susan model. I think her name is Susan. Desk job model. I think her name's Susan. It might not be. No. Susan. There's not Susan. Doll desk job model. No. Maybe? No. Where is it? Okay, here it is. Go away. CDC, here we go. Workplace health model. No, that's not what I wanted. Here we go. So this lady, what is her name? So this is a model that Emma, her name's Emma, not Susan. Prominently hunchback, varicose veins, red eyes, and protruding stomach. So they basically took what health effects they know can be caused by desk jobs, and they took them to the extreme. This hunched back, the neck, lower neck protrusion. She's got swollen wrists, swollen ankles, bloated gut, and her skin's really gross. In just 20 years' time, the average office worker will have a hunched back, protruding stomach, and sore eyes, according to a life-size doll developed as part of a report into workplace health. So yeah, they made her to kind of show what's going on. And that's not very good. That's not. Her back makes my back hurt. Yeah. Anyways, yeah. Emma. Okay, moving on from the sad state of things in our reality, tooth study prompts rethink of human evolution. A study into tooth wear in a group of wild Japanese macaques have significant implications for the study of human evolution, a University of Otago study has shown. Lead author, studied root grooves and large uniform scratches into the macaque's teeth, which had previously only been described in fossil humans. Unusual wear on our fossil ancestors' teeth is thought to be unique to humans and demonstrates specific types of tool use. These types of wear have also been considered some of the earliest evidence of cultural habits for our ancestors. However, our research suggests this idea may need reconsidering, since we describe identical tooth wear in a group of wild monkeys that do not use tools. This research raises questions for our understanding of cultural changes during human evolution and suggests we may need to reassess early evidence of cultural habits. Okay, what was this? Oh, toothpick. Oh, so toothpick-like grooves on back teeth and large uniform scratches on the macaque's front teeth were actually caused by something more mundane, yet still surprising: eating shellfish from rocks and accidentally chewing grit and sand with their food. This macaque group is well known for undertaking remarkable behaviors, including washing foods in water and consuming fish. They have been studied for more than 70 years and have not been seen using tools or other items that could cause the unusual tooth wear observed. Pathologies in a wide variety of primate species, and was extremely surprised to find this type of tooth wear in a group of wild monkeys. Up until now, the large scratches in the front teeth of fossil humans have been considered to be caused by a behavior called stuff and cut, in which an item such as an animal hide is held between the front teeth and a stone tool is used for slicing. Similarly, toothpick grooves are thought to be caused by tools being placed between back teeth to remove food debris or relieve pain. Although this does not mean hominins were not placing tools in their mouths, or studies suggest the accidental ingestion of grit and or normal food processing behaviors could also be responsible for this atypical wear pattern. We are so used to trying to prove that humans are unique that similarities with other primates are often neglected. Studying living primates today may offer crucial clues that have been overlooked in the past. Interesting. Okay, cool. So that, not saying it couldn't be from toothpicks and tool use, but it could not be too. That's kind of important. Alright, last one for today. How bones communicate with the rest of the body. Bones, they hold us upright, protect our innards, allow us to move our limbs, and generally keep us from collapsing into a fleshy puddle on the floor. When we're young, they grow with us and easily heal with playground fractures. When we're old, they tend to weaken and may break after a fall or even require mechanical replacement. If that structural role was all that bones did for us, it would be plenty. But it's not. Our bones also provide a handy storage site for calcium and phosphorus, minerals essential to nerves and cells to work properly, and each day their spongy interior, the marrow, churns out hundreds of billions of blood cells, which carry oxygen, fight infections, and clot the blood in wounds, as well as other cells that make up cartilage and fat. Human bodies are fascinating. Even that's not all they do. Over the past couple of decades, scientists have discovered that bones are participants in complex chemical conversations with other parts of the body, including the kidneys and the brain, fat and muscle tissue, and even microbes in our bellies. It's as if you suddenly found out that the studs and rafters in your house were communicating with your toaster. Scientists are still deciphering all the ways that bone cells can signal other organs, and how they interpret and respond to molecular messages coming from elsewhere. Already, physician-scientists are starting to consider how they might take advantage of these cellular conversations to develop new treatments to protect or strengthen bone. Ooh, that'd be cool. Man, Granny doesn't have to fall and break her hip anymore. We're good. We're going to strengthen those bones. That'd be so awesome. Could also help with the healing process after a fracture. It could occur faster or something. It's a whole new era of exploration, says Laura McCabe, physiologist at Michigan State University in East Lansing. Early evidence that bone has something to say. Bone is a unique tissue. It contains not only cells that build the hard matrix that gives the skeleton its strength, but also cells that break it down, enabling bone to reshape itself as a child grows and to repair itself throughout life. The bone builders are called osteoblasts, and the disassembly crew consists of cells known as osteoclasts. When the balance between the actions of the two is off-kilter, the result is too little or too much bone. This happens, for example, in osteoporosis, a common condition of weak and brittle bones that results when bone synthesis fails to keep up with degradation of old bone. In addition to osteoblasts and osteoclasts, bone contains another cell type, the osteocytes. While these cells comprise 90% or more of bone cells, they weren't studied much until about 20 years ago, when a cell biologist named Linda Bonewald what an awesome name for this, got interested. Colleagues told her not to waste her time, suggesting that osteocysts probably only played some mundane role, like sensing mechanical forces to regulate bone remodeling, or maybe they were just kind of there, not doing much of anything. Man, don't ever stop people from questioning. Why? Oh, they probably do this. So let's prove that that's what they do. Bonewald, now at India University in Indianapolis, decided to investigate them anyway. You go, girl. Osteocytes do, in fact, sense mechanical load, as she and other researchers have found. But, as Bonewald says, they do so much more. She recently wrote about the importance of osteocytes to the kidneys, pancreas, and muscles. So, you got these. The osteoblast builds bone matrix. The osteoclast sorry, I smelled blood, so I was afraid I was getting a bloody nose. Breaks down bone matrix, and then osteocyte maintains bone and communicates with other organs. The first finding regarding osteocyte communication with other organs reported in 2006 was that the cells make a growth factor called FGF23. Don't you love these names? The molecule then cruises the bloodstream to the kidneys. If the body has too much FGF23, as happens in an inherited form of rickets, the kidneys release too much phosphorus into urine, and the body starts to run out of the essential mineral. The resulting symptoms include softened bones, weak or stiff muscles, and dental problems. Around the same time that Bonewald was diving into osteocyte research, physiologist Gerard Carcenti began investigating a potential relationship between bone remodeling and energy metabolism. Carcenti, now at Columbia University in New York, suspected that the two would be related because destroying and recreating bone is an energy-intensive process. In a 2000 study, Carcenti investigated whether a hormone called leptin could be a link between these two biological processes. Leptin is produced by fat cells and is best known as a depressor of appetite. It also emerged in evolution around the same time as bone. In experiments with mice, Carcenti found that leptin's effects in the brain put the brakes on bone remodeling. Using leptin in this way, Carcenti suggests would have allowed the earliest bony creatures to suppress bone growth alongside appetite when food was scarce, saving their energy for day-to-day functions. His group found support for this idea when they took x-rays of the hand and wrist bones of several children who lack fat cells, and thus leptin, due to a genetic mutation. In every case, radiologists unfamiliar with the people's true ages ranked the bones as months or years older than they were. Without leptin, their bones had sped ahead, acquiring characteristics like higher density that are more typical of older bones. That was a case of bone listening to other organs, but in 2007, Carcenti proposed that bone also has something to say about how the body uses energy. He found that mice lacking a bone-made protein called osteocalcin had trouble regulating their blood sugar levels. In further research, Carcenti discovered that osteocalcin also promotes male fertility via its effects on sex hormone production, improves learning and memory by altering neurotransmitter levels in the brain, and boosts muscle function during exercise. He described these messages and other conversations that bone participates in the annual review of physiology. It's a spectacular set of functions for one molecule to handle, and Carcenti thinks they're all linked to a stress response that early vertebrates, animals with backbones, evolved for survival. Bone may be an organ defining a physiology of danger, he says. Carcenti proposes that osteocalcin's effects allowed early vertebrates, both male and female, to respond to the sight of a predator by amping up energy levels through the effects of testosterone as well as muscle function. They'd be able to run away and later remember and avoid the place where they'd encountered that threat. Researchers in Carcenti's lab did these studies with genetically modified osteocalcin-deficient mice that he developed, and several labs have replicated his results in various ways. However, labs in the US and Japan working with different strains of mice that don't make osteocalcin didn't see the same widespread effects on fertility, sugar processing, or muscle mass. The scientists haven't yet been able to explain the disparities, and the danger response hypothesis remains somewhat controversial. Whether or not osteocalcin played the big role in vertebrate evolution that Carcenti proposes, these studies have inspired other scientists to examine all kinds of ways that bone listens to and talks to the rest of the body. Crosstalk between muscle and bone. Bone and muscle, partners in movement, have long been known to interact physically. Muscle tug on bone, and as muscles get stronger and larger, bone responds to this increased physical pull by becoming bigger and stronger too. That allows bone to adapt to an animal's physical needs, so the proportional muscle and bone can continue to work together effectively. But it turns out that there's also chemical conversation going on. For example, skeletal muscle cells make a protein called myostatin that keeps them from growing too large. In experiments with rodents, alongside observations of people, researchers have found that myostatin also keeps bone mass in check. During exercise, muscles also make a molecule called beta-aminoisobutyric acid. Beta-aminoisobutyric acid. BABA. That influences fat and insulin responses to the increased energy use. Bonewall has found that BABA protects osteocytes from dangerous byproducts of cellular metabolism, called reactive oxygen species, in young mice that were immobilized, which normally causes atrophy of bone and muscle. Providing extra BABA kept both bones and muscle healthy. Ooh, that's interesting. That could be really useful for, astronauts to help, because they tend to have a lot of atrophy going on being in space for long periods. If that could help, probably not replace the use of actual exercising, but could aid it, and maybe people could stay in space longer. That'd be cool. Or it could help with people who are unable to move for an extended period of time, or permanently unable to move, and people in a coma or something, that'd be awesome. How bone talks with the rest of the body. In additional studies, Bonewald and colleagues found that another muscle molecule that increases with exercise, irisin, also helps osteocytes to stay alive in culture and promotes bone remodeling in intact animals. The conversation isn't all one way either. In return, osteocytes make prostaglandin E2, which promotes muscle growth. On a regular basis, they boost production of this molecular messenger when they experience an increase in the tug from working muscles. What bone gets from the gut? The human body contains about as many microbial cells as human ones. In the trillions of bacteria and other microorganisms inhabiting the gut, its microbiome function almost like another organ. They help to digest food and prevent bad bacteria from taking hold, and they talk to other organs, including bone. So far, the bone microbiome conversation seems to be one way. No one has observed bone sending messages back to the microbes, says Christopher Hernandez, a biomechanics expert at Cornell University in Ithaca, New York. But the skeleton can learn a lot of useful things from the gut, McCabe says. For example, suppose a person gets a nasty case of food poisoning. They need all their resources to fight off the infection. It's not the time to build a bone. The first hints of bone microbiome connection came from a 2012 study of mice raised in a sterile environment. without any microbes at all. These animals had fewer bone-destroying osteoclasts and thus higher bone mass, giving the mice a full complement of gut microbes, restored bone mass to normal in the short term. Ooh, weird. But the long-term effect was a bit different. The microbes released molecules called short-chain fatty acids that caused the liver and fat cells to make more of a growth factor called IGF-1, which promoted bone growth. Gut microbes also appear to moderate another signal that affects bone, parathyroid hormone, PTH, from the parathyroid glands at the base of the neck. PTH regulates both bone production and breakdown, but PTH can only promote bone growth if mice have a gut full of microbes. Specifically, the microbes make a short-chain fatty acid called butyrate, and that facilitates this particular conversation. Incidentally, that FGF23 made by osteocytes also acts on a parathyroid glands, turning down their secretion of PTH. While scientists have uncovered many important roles for the gut microbiome in recent years, it wasn't a given that they'd influence the skeleton, says Bonewald. Boy, were we surprised to see effects on bone. Yeah, so your gut microbiome is so important. It talks to your entire body. You need to keep it healthy, and to do that, you regulate what you're eating. So eating a shit diet, you're gonna affect down to your bones. It's important. Eat well. Can doctors join the conversation? They should. The most thrilling thing about these organ-to-organ messages, says McCabe, is that it suggests novel ways to help bone with medicines that act in different parts of the body. We could be even more creative therapeutically, she says. The Centers for Disease Control and Prevention estimates that nearly 13% of Americans over 50 suffer osteoporosis. This could actually be a really more natural way of fighting these illnesses and these degenerative things that people go through. And while there are several medications that slow the breakdown of bone, as well as some of the speed build up, they can have side effects, and they're not used nearly as much as they could be. An endocrinologist at the Mayo Clinic in Rochester, Minnesota, that's why he says new approaches are needed. One obvious place to start is with the gut. Probiotics and other foods containing cultured microbes such as the fermented milk, kefir, can help build a healthy microbiome. Yeah, I don't know if just taking pills is going to help. Taking pills of probiotics isn't going to offset the shitty diet that you have. McCabe's group found that a particular probiotic bacterium that normally follows antibiotic treatment, another group tried a combination of 3 types of lactobacillus in postmenopausal women, the segment of the population most susceptible to osteoporosis, and those on the treatment experienced no bone loss during the year-long study, whereas those in a placebo group did. Hernandez has been investigating another therapeutic approach that would improve bone's resilience, but not by adding mass or preventing breakdown. The work grew out of a series of experiments in which he used antibiotics to perturb, but not eliminate the gut microbiome in mice. He predicted that this would cause the mice to lose bone mass, but the results surprised him. It didn't change the density or the size of the bone, he says, but it changed how strong the bone was. The bones of the antibiotic-treated animals were weak and brittle. Investigating further, Hernandez's team found that when mice receive antibiotics, their gut bacteria stop making as much vitamin K as they normally do, and so less of the vitamin reaches the large intestine, liver, and kidneys. The result is alterations to the precise shape of mineral crystals in the bone. Hernandez is now investigating whether the source of the vitamin K, either from gut microbes or dietary sources like leafy greens, matters for bone crystallization. If people need the bacterial version, then probiotics or even fecal transplants might help, he suggests. Ugh, no. probiotics and a good diet. No. Crescenti's work, meanwhile, has inspired an entirely different strategy as he observed early on. Leptin from fat cells slows bone formation via the brain. In response to leptin, the brain sends a signal that ultimately activates bone cells' beta-adrenergic receptors, shutting off bone-building osteoblasts and stimulating bone-clearing osteoclasts. These are same beta-adrenergic, that's a weird word, receptors exist in various parts of the body, including the heart, and drugs that block them are commonly used to reduce blood pressure. To investigate whether these drugs might also prevent osteoporosis, Khosla tested a few different beta blockers in 155 postmenopausal women, and 2 of the drugs seemed to keep bones strong. He is now running a larger study with 420 women. Half will receive one of those drugs, atenolol, and the other half will get a placebo for 2 years. The scientists will monitor them for changes in bone density in the hip and lower spine. Khosla has another idea based on the fact that as bone ages, it accumulates old senescent osteocytes that produce inflammation. The inflammation in turn can affect the constant buildup and breakdown of bone, contributing to their imbalance in osteoporosis. Cyanolytics are drugs that cause those old cells to kill themselves, and Khosla recently co-authored a summary of their potential for the Annual Review of Pharmacology and Toxicology. In a study in older mice, for example, this kind of medication boosted bone mass and strength. Khosla has another trial going with 120 women aged 70 or older to test the ability of senolytics to increase bone growth or minimize its destruction. Scientists will have plenty to learn about the conversations between bone and the rest of the body. With time, this research may lead to more therapies to keep not just the skeleton, but also the other conversationalists healthy and strong. That's so... Yes, let's do it! For the grannies! But what's clear already is that the skeleton is not just a nice set of mechanical supports. Bones constantly remodel themselves in response to the body's needs, and they're in constant communication with other parts of the body. Bone is a busy tissue with broad influence, and it's working behind the scenes during the most basic daily activities. So the next time you enjoy a cup of yogurt, work out, or even empty your bladder, be sure to spare a moment to thank your bones for responding to microbial signals, conversing with your muscles, and keeping your phosphorus supplies from going down the drain. That was a cute little ending. Anyways, that's about it. That's the last article. But I think I remembered having a couple Twitter things to share. I am on Twitter. You can follow me. Actually, I think it's Amber Bones. Yeah, at Amber_Bones. I follow a lot of other anthropologists, archaeologists, and paleoanthropologists. Science Twitter is pretty active. There's a few specific researchers that I quite enjoy. Lee Berger is one of them. He really is very active about bringing in people and just being active on social media and talking about his excavations as he's doing it. A little bit ago, he actually took a camcorder into the Naledi cave system at Rising Star so that people could go in and see what's going on. It's amazing. I really actually quite like that, just being really open about things, because I think the day-to-day logistics of archaeology and paleoanthropology, in his case, aren't really well known, and all the little things that you have to go through to get that one little fossil out is really interesting. So yeah, give me a follow at Amber_Bones on Twitter. and let's enjoy some cool archaeology together. Right here we have this mysterious Lascaux scene that was created down a deep natural shaft. Has the wounded bison attacked its hunter? Why is the man drawn so simply compared with the animals? What is the bird stick? How does the rhino relate? Is this even a composition painting at one time? So here we have a large bison, I think they called it. And then over here, a rhino, you can tell from the horns here. Although, I probably would have guessed pig, but it's a rhino. And then you have a man here, and he put man in quotes, but I'm not sure if it's a quotable thing. Then you have like a bird-shaped thing on top of a stick. I'm not a cave painting expert, but I would say that these were done in 2 different times. The style here, how thick the strokes are way different than this guy and this. Like, this, it's so thin and different. It just, I don't know. Seems like 2 different ones. Anyways, so that's interesting. Cave drawings. Gotta love them. Next. Oh, not that. That's. Alright. Hystera Oase, Romania, is where cave explorers found a deep ancient cave bear hibernaculum with remains of 2 humans between 42,000 and 37,000 years old. These people had more Neanderthal ancestry than anyone alive today. So, this cave system, look at all this. They had to wire in lights, they have to wear suits, caving things. Look at this. I would love to be in there. I really would. Awesome. Alright, even small bits of the even small bits of the Mesolithic are simply stunning. Flake core from the Microbiome culture archaeology. So this is a core. It's just a good chunk of material chert or obsidian or something that you would take pieces off, and those pieces are actually what the tool is going to be. It's not the core. So you can actually tell they struck up here, and then this sliver came off, this shard came off, and then you have another one right here where they struck right here, breaking this one off. Over here they struck up here, and it would have broken this portion off. So that's a very beautiful core. Indeed. I've seen some really gnarly cores out there before. That's a very nice and clear core. UW 101-1400 is part of a lower jaw of an infant Homo naledi. Is more of this poor Pleistocene child waiting to be found in Rising Star Cave. So you can see the tooth development going on. So we have our first molar right here, hasn't yet erupted. You have a canine, maybe the not the canine, the first premolar, the first premolar here, and then you have your second and first molars, the infant molars, your canine here. You got a tooth forming right here, which is probably actually that's probably the canine, and that's probably a premolar. I don't know. But yeah, it's forming all in here. You scanned it and it's all hidden in there. Beautiful. A nice meme for you, publishing scientific papers is like gambling, a reviewer one. Your manuscript is really interesting and well written, but reviewer too. Oh no, it's not. Oh man. Gotta love the memes. Okay. What else we got? Old fieldwork photos, the Chilean bearing excavation, one from Wonderwork Cave, South Africa. You can see rock art on the left well as well, part of the beautiful rounded phreatic tunnel that runs about 140 meters into the Dolomite hillside. So, let's see. Oh, yep, you see right here we have the drawings going on. Nice hole there. So this is an active archaeological site. When you're doing a full excavation like this, you're going to measure out your meter by meter unit. You're going to put up a grid, keep it aligned as best as possible, and that's how you keep things organized and controlled. Otherwise you're just going all over the place. And when you have this grid system put up, you can even see, so over here they went much deeper, but they have the strings coming down to help indicate where that unit is, even down there. It helps you with mapping, because you actually have to map the layers as you excavate them. So you draw what you see, you take a photo of what you see, and then you're able to remove the artifact, trying to capture as much data as you possibly can before removing it. Alright, last one. Not anthropologically related, but a baby dinosaur. Look at it. Look at the little baby dinosaur. It's adorable. Anyways, we're done. Oh, even for scale, a finger. It's like using the banana for scale. You can't really tell because fingers are so... I got small hands compared to other people. I'm sure, like, Shaq could just, like, ball my head up with one hand, you know? Anyways. A ruler would have been better than that. It's a really weird scale thing. Anyways, that's all I have for you guys today. I hope you enjoyed the stream. I hope to see you guys next week. I should be on time next week. I don't have a meeting, hopefully. If there are any changes, I post it on Twitter. So follow me at Amber_Bones, and then give the Discord a follow as well. Join it so that we can talk. I watch movies sometimes. We do documentaries and such. I also post the links for all of the articles I cover on stream. So if you want to do any more research on your own, it's that. And yeah, I hope you guys have a wonderful evening. Take care.